We're trying to help you understand how scientists use DNA to uniquely identify an individual in a forensics investigation. Doing some DNA math will not only help you understand where we are going to look in the genome for unique identifiers, but also how it's possible to use DNA technology to find these small regions of variability. The first step is to figure out how much of our DNA is in the genes where we don't expect to find variability. There are about 3 billion bases in the human genome. That's 3 times 10 to the 9th in scientific notation. The human genome contains approximately 30,000 genes, and an average gene contains about 1,000 bases. Knowing these values, what percent of the total DNA is in our genes? If each gene has 1,000 bases and there are 30,000 genes, then 1,000, 1 times 10 to the 3rd, times 30,000, 3 times 10 to the 4th, is the number of bases contained in the genes. The total number of bases in the genome is 3 times 10 to the 9th, 3 billion. To calculate the percent of the total DNA that is in our genes, you need to divide the number of bases in the genes by the number of bases in the genome. Percent is part divided by whole. The percent of total DNA contained within the genes would be 3 times 10 to the 7th divided by 3 times 10 to the 9th equals 1 times 10 to the negative 2, which is 0.01 or 1%. Remember, you need to add exponents when you multiply and subtract exponents when you divide. So only about 1% of human DNA is contained within genes, and these regions contain very little variability since this DNA codes for protein. In a crime scene investigation, we are trying to identify one individual whose DNA is the only DNA that matches that obtained at the scene of the crime. What makes DNA unique to a single individual? It's regions that show differences among all humans, or what are called variable regions. These regions are likely to be in the 99% of DNA that is outside of the genes, the intergenic DNA. Interestingly, intergenic DNA is 99% identical between individuals, and only 1% is variable. But even this 1% is a large amount of DNA, about 30 million bases. Remember, we have 3 billion bases in our total genome. So these two facts are what allow us to do DNA forensics. One, that 1% of our DNA, 30 million bases, is variable, allowing it to be unique in a single individual. And two, that 99% of our DNA is identical, so we can take advantage of the polymerase chain reaction, or PCR. You'll learn more about how PCR works in the next section.